My name is Jacob Henson, and I will be discussing DNA replication of a prokaryotic organism. I'd like to start out by laying out our key players in replication. Initiator region and protein. This is where the DNA will start to unravel and replication will begin. Helicase assists in breaking phosphodiester bonds so that double strand can separate. Single-stranded binding protein binds to keep the DNA in organized fashion. Ligase seals the nick in the double-stranded molecule at the completion of replication. RNA polymerase or primase synthesizes primers. DNA polymerase 1 essential in the replacing of RNA primers with actual DNA nucleotides. And DNA polymerase 3 works in replication, deleting and lagging strands. Now that we know what types of components we're working with, we can begin talking about how prokaryotic cells replicate. Let's take a bacterial cell like Streptococcus and show how it would replicate its own DNA in a normal environment. The fascinating thing about prokaryotic DNA is it has circular chromosomes, so these cells will replicate in what is known as rolling circle. Here I have a diagram that I've drawn that shows all the different enzymes and proteins that work to make replication possible. Here we have our leading strand on the bottom that is replicating in the 3 to 5 direction. This strand is a continuous line into the replication fork. As you can see, DNA polymerase 3 is adding nucleotides. The single-stranded bonding proteins keep the DNA organized and prevent it from winding up. If you look to the right of the diagram, you can see helicase as it splits the DNA so it can be replicated. Now we look at the top side of the diagram. Replication is occurring in the 5 to 3 direction. There's a lot more going on. We see a few more proteins and enzymes in action. We have RNA polymerase, also known as primase, which is making RNA primers to begin replication in these certain places. And to the left of that, we see DNA polymerase 3, which is adding nucleotides and replicating DNA. Then we see DNA polymerase 1, which is replacing these RNA primers with DNA nucleotides. On the lagging strand, we have portions of DNA called Okazaki fragments, which make it discontinuous. But these fragments are sealed up with ligase at the very end. Here we have a diagram of rolling circle replication. I want to point out how replication will follow the same pathway as the diagram we looked at before, but the orientation is different. I visualize this type of replication as if I'm unrolling toilet paper, and at the very end, when the toilet paper is torn, is comparable to DNA replication. The initiator protein binds at their origin. A nick in the strand occurs, and the protein binds onto the 5' end. The 3' end serves as the primer for DNA polymerase 3. And the same process occurs as the diagram that we just looked at. Well, here we can observe 2 ends meeting, the binding protein sloughing off, and the ends bind. A loop in the single strand occurs and allows RNA polymerase to make primers, and then the replication process continues. Finally, DNA ligase seals the nick, and we have our new DNA molecule. If there is an absence of DNA polymerase 1, there will be no enzyme to proofread to ensure bases are paired correctly, as well as no way for the cell to replace RNA primers with DNA. There will be an abundance of RNA primers and no actual replication occurring.